Assalamualaikum and hi everyone. So I hope everyone are in good condition. So this is the subtopic 11.4 inheritance in human from chapter 11 inheritance. So this is the last video from chapter 11. Okay, in this subtopic we will learn about the inheritance in humans. So first of all, we have to learn about the types of chromosomes. Okay, there are two types of chromosome, human chromosome, namely autosome and sex chromosome. So human somatic cell consists of 44 autosomes and 2 sex chromosomes. Autosomes vary in terms of size and length. So this is the picture of the autosomes and sex chromosome. Okay, they consist of 44 autosomes which arranged in pair, 22 pairs. That means 44 autosomes, and this is 2 sex chromosome. Okay, as the sex chromosome is either in X and Y chromosome. So this is the differences between the feature, function and example of autosome and sex chromosome. Okay, so the feature, autosome consists of chromosome pairs from number 1 to number 22, while the sex chromosome consists of 1 chromosome pair, that is number 23. The function of autosome is control or characteristic of somatic cells, but sex chromosome consists of genes that determine the gender. So example, the type of chromosome is example of autosome is the types of blood group, height and skin color. Okay, the sex chromosome, the example of sex chromosome, male has XY chromosomes. Okay, male has XY sex chromosome, while female has XX sex chromosomes. So this the karyotype, and the karyotype is the number and the structure in chromosome present in a cell nucleus. Okay, so the karyotype, the arrangement of chromosome, okay, in pairs based on the homologous chromosome in terms of their size, centromere location and bending pattern of a chromosome. So this is the male karyotype, and for male. Okay, the structure, the karyotype is the arrangement of homologous chromosome based on their size, based on the centromere location and bending pattern. So this is for female karyotype. Okay, male karyotype, we can write down the male karyotype as 44 plus XY. For female karyotype, we can write as a 44 plus XX. 44 means autosomes, XY, or XX means sex chromosome. Okay, so there are change in number of chromosome can occur due to failure of homologous chromosome to separate during anaphase 1, or failure of sister chromatic to separate during anaphase 2. This disorder is known as Nondistension, which can occur in some chromosomes. When non-disjunction occur in human, either male gamete sperm or female gamete ovum can possess chromosome number of less than 23, that is 22, or more than 23, which is 24. Therefore, fertilization that involves the abnormal gamete, which a normal gamete produce a zygote with 44 chromosome or 47 chromosome. So the example of genetic disease caused by the non-disjunction are Down syndrome, Turner syndrome and Klinefelter syndrome. So this is the Down syndrome, karyotype of Down syndrome. This is the karyotype of Turner syndrome. This is the karyotype of Klinefelter syndrome. Okey, jadi sebenarnya perubahan yang berlaku pada genetic disease ni disebabkan oleh kegagalan homologous chromosome tu berpisah semasa anaphase 1, iaitu semasa meiosis parent. Okay, anaphase 1 ataupun kegagalan sister kromatik dia berpisah pada anaphase 2. Jadi keadaan ni dipanggil non-disjunction. Bila berlaku kegagalan untuk berpisah kromosom ni, dia akan menyebabkan gamet yang dihasilkan oleh parent tu sama ada kurang kromosom ataupun terlebih kromosom. Okey, bila kurang kromosom, kalau fertilization berlaku yang melibatkan abnormal gamet ni, dia akan menghasilkan zygote yang mempunyai terlebih kromosom atau mungkin mengalami kekurangan kromosom. Contoh kalau Down syndrome ada 47 kromosom. Okey. Kalau Turner syndrome ada 45. So this is the non-disjunction. Okay, this is the homologous chromosome fail to separate during anaphase 1. Okay, or non-disjunction in meiosis 2, anaphase 2. This is the sister chromatid that fail to separate. So normal meiotic division, it produce half the number of chromosome, but for non-disjunction, it will produce either the chromosome have more number or less number. So this is the karyotype of a male with Down syndrome. Okay, total chromosome number in Down syndrome is 47, which is karyotype is 45 plus XY. There is an extra chromosome for chromosome pair in number 21. This is the extra number. So the Down syndrome is also known as trisomy 21 because trisomy means 3 and 3 chromosome at number 21. So Down syndrome can occur in both males and females. So this is the Turner syndrome. This is the karyotype of Turner syndrome. In Turner syndrome, total number of chromosome is 45, which is the karyotype is 44 plus XO. So a missing X chromosome in a pair of sex chromosome. The gender of the individual Turner syndrome is a female. So here you can see here the sex chromosome is only 1. Okay, so 45 means 44 autosome plus X only. So this is the Klinefelter syndrome karyotype. So the karyotype of Klinefelter syndrome has total number of 47, but extra in X chromosome. Okay, there is an extra of X chromosome in a pair of sex chromosome. The gender of individual with Klinefelter syndrome is male. However, his secondary sex characteristics are not well developed. Okay, so we can see here extra number of X chromosome. Okay, the male with the extra number of X chromosome. So let's discuss more about the human inheritance. So in human inheritance, first of all, we learn about the ABO blood group. How the blood group in human is determined. So ABO blood group human is classified into blood group A, blood group B, blood group AB and O blood group. So ABO blood group in human is an example of multiple allele. Okay, what's mean multiple allele? Okay, multiple allele is more than a pair of allele. Okay, before this we have learned about one characteristic is controlled by a pair of allele. Okay, but for ABO blood group is multiple allele. So that's mean there are 3 allele: allele IA, allele IB and allele IO. Here. Okay, so that this allele determine the type of integer. Okay, presence of the surface membrane of the red blood cell. So look at the picture. So this is the diagram to show the antigen found on the surface of the red blood cell. Okay. However, a person only possesses 2 allele to determine his or her blood group. Okay, jadi walaupun multiple allele, tetapi seorang seseorang individu tu mempunyai 2 allele saja untuk determine dia punya blood group ataupun nak menentukan blood group, disebabkan kita ada satu homologous chromosome kita dalam bentuk sepasang, jadi dia akan ada 2 allele saja. So banyak alternatif yang akan hasil. That's why phenotype of blood group dia ada 4 jenis. Okay, so both IA and IB are dominant allele, whereas the IO is recessive. Okay, and therefore a combination of IA and IO allele express a blood group phenotype, whereas IB IO express a blood group B blood phenotype. So IA IB allele are codominant to one another. When these 2 alleles are present together, effect of both alleles show. A combination of both alleles give a AB blood group phenotype. So for blood group A, okay, person who have blood group A, the genotype is either IA or IAIO. Okay, sama ada the homozygote dominant or heterozygote. Blood group B, IB or IBIO. Okay, dominant recessive, dominant jugalah. Blood group B. Blood group AB is codominant. Combination between the LLIA and LLIB. Okay, but blood group O must combine between the LLIO and LLIO. So this is the example of the inheritance in human. A man with a blood group married with a woman with B blood group. Explain the probability of a couple is getting a child with O blood group. Okay, macam mana lelaki yang mempunyai blood kumpulan darah A berkahwin dengan perempuan yang mempunyai kumpulan darah B, apa dia punya kebarangkalian untuk mendapatkan anak dalam blood group kumpulan darah O. Okay, so both mother and father are heterozygote, baru akan dapat anak blood group O. Okay, so father receive IAIO, mother IBIO. Kalau father IAIA dengan mother IBIB, dia tak akan dapat blood group O. Jadi heterozygotes kita kena melibatkan heterozygote. So father, okay, this is the genotype father. So father will produce gamete. Okay, so this is the gamete of father, IAIO, mother IBIO. After random fertilization, the genotype, okay, IAIB, IAIO, IBIO and IOIO. So this is the probability. Okay, phenotype, okay, AB, A, B and O. Phenotypic ratio is 1:1:1:1. So the percentage for getting the blood group O for this couple is 25%. Okay, 25%. 1 daripada 4. 1/4. 25% anak dia akan mempunyai kumpulan darah O. Jadi kalau blood group father, kalau kita tulis sebagai homozygote dominant, IA kahwin dengan ibu IB, dia tak akan dapat darah anak yang mempunyai kumpulan darah O. Dia akan dapat anak mempunyai kumpulan darah AB sahaja. Okay, another blood group is called Rhesus factor. So Rhesus factor, beside the antigen A and antigen B, we have learned previously. Antigen A and antigen B is a type of antigen found on the surface, but there is another antigen called antigen D, known as Rhesus factor. So we can see here, this is the antigen. Okay, antigen Rhesus D factor. Okay, so the Rhesus factor is called antigen D. An individual whose red blood cell has Rhesus factor is said to be Rhesus positive, while an individual without the Rhesus factor is said to be Rhesus negative. Okay, Rhesus positive maksudnya ada antigen D. Rhesus negatif tiada antigen D. Okay, so to write down the genotype, Rhesus positive either RH positive, RH positive, or RH positive, RH negative. For the Rhesus negative, RH negative, RH negative. So inheritance of Rhesus factor from the parent to the children is based on the principle of Mendel's law. Rhesus factor is controlled by genes that consist of a pair of allele, namely Rhesus positive dominant, Rhesus negative is recessive. Rhesus positive is dominant, Rhesus negative is recessive. So Rhesus positive individual either homozygotes dominant RH plus and RH plus ataupun heterozygote RH plus or RH negative. Okay, RH positive, RH negative. So RH negative individual is homozygote recessive. Okay, RH negative, RH negative. So this is the situation. Okay, inheritance in Rhesus. Okay, example, father have Rhesus positive. Okay, Rhesus positive, mother have Rhesus negative. So, and when meiosis, father will produce gamete contain allele Rhesus positive and allele Rhesus negative. So, fertilization, we produce RH positive, RH negative, which means phenotype is Rhesus positive, same as father. So, the ratio is all children are Rhesus positive. Okay, another situation, when the father is heterozygote, RH positive, RH negative genotype, and the genotype of mother is RH negative and RH negative. So father will produce two types of gamete that carry allele Rhesus positive and Rhesus negative, and mother will carry gamete with allele RH negative. So when random fertilization occur, okay, it will produce 50% Rhesus positive, okay, and 50% Rhesus negative. So half 1/2 Rhesus positive, okay, and 1/2 Rhesus negative. So for this couple, 50% their children is Rhesus positive and 50% Rhesus negative. So this is the location of a gene. Okay, ABO blood group. ABO blood group we can see here at the chromosome number 9. Okay, so we have the Rhesus blood type. Okay, Rhesus blood type we can found on the chromosome number 1 here. Okay, so we will discuss about the disease related to the inheritance. Okay, which is thalassemia. Okay, how the thalassemia are inherited. Thalassemia is an inherited disease. The disease can pass down from generation to generation. Thalassemia is due to the gene mutation. Okay, later we will learn about the mutation on chapter 12 on the autosome, and that is on chromosome 11 or 16. So thalassemia is due to the abnormally and low number of hemoglobin. So the red blood cell is smaller and paler. So this is the picture of thalassemia. Okay, the abnormality and the low number of hemoglobin. So thalassemia is caused by the gene mutation at the chromosome number 11 or number 16. So this is the example, chromosome number 11. So this is normal. So when the mutation occurs, so the person have maybe carrier or thalassemia minor or the symptomatic thalassemia. Okay, and then this the hemoglobin part hydroxylase. Ini the severe. Okay, this is the situation of the inherited disease. Inheritance of thalassemia. Thalassemia carrier is said to have thalassemia minor condition in which the individual possesses recessive allele of thalassemia, but the individual does not show any symptom of the disease. So maksudnya bila father carrier, ayah carrier, mak normal, so dia akan menghasilkan 50% anak yang normal dan 50% anak yang carrier. Carrier ni maksudnya thalassemia minor. Dia tidak menunjukkan simptom. Dia cuma carrier pada recessive allele. Okay, tapi jika pasangan kedua-duanya carrier, okay, dia akan mendapat 50% anak dia carrier dan 25% normal, dan ada anak yang akan thalassemia. Okay, 25% anak akan thalassemia. Okay, so detection of thalassemia can only be confirmed by a blood test. So a thalassemia patient is said to have thalassemia major when the individual have both recessive allele. Okay, jadi thalassemia ni akan dapat bila dia dapat kedua-dua recessive allele daripada father dan mother. So thalassemia patient show symptom such as tiredness, paleness, breathing difficulty and changes in facial bone formation from age 3 to 18 months. Okay, so yang thalassemia ni sahaja yang akan dapat simptom. Okay, kalau carrier tak ada simptom untuk carrier. Okay, next we will learn about the sex determination. Okay, penentuan jantina. Lelaki dan perempuan dalam anak-anak. A male has 44 plus XY chromosome and a female has 44 plus XX. So this is the human karyotype we have learned previously. So sperm produced in the testes are haploid, and each sperm has either 22 plus X or 22 plus Y chromosome. Okay, but the secondary oocyte produced in ovary also have haploid, and each secondary oocyte only one set of chromosome, namely 22 plus X chromosome. So sex and gender of a child is determined during fertilization. So maksudnya sekarang ni mother, okay, this is the karyotype of mother, female, 44 plus XX. So mother akan produce ovum 22 plus X only, one set. But father 44 plus XY. So after meiosis, father akan produce dua jenis sperm, iaitu satu 22 plus X ataupun 22 plus Y. So this is the example of sex determination, inheritance. So father produce 44 plus XY, mother produce 44 plus XX. Okay, the genotype of father 44 plus XY, the genotype of mother is 44 plus XX. After meiosis, father will produce sperm, okay, 22 plus X or 22 plus Y. Mother will produce only one set, 22 plus X. So after when the random fertilization occur, if the 22 plus X fertilize with 22 plus X, it will produce 44 plus XX. It means female. Okay, if the sperm with 22 plus Y fertilize with the ovum or secondary oocyte 22 plus X, it will produce 44 plus XY, which means male. So the phenotypic ratio is 1:1. 50% the couple getting female daughter, female child, of 50% have male. Okay, son. Okay, 50% have son or 50% daughter. Okay. Now sex link inheritance. Okay, sex link inheritance. The genes located on the sex chromosome which control specific characteristic but are not involved in sex determination are known as a sex link genes. Okay, so genes of color blindness and hemophilia are located in X chromosome. So the color blindness and hemophilia is the example of sex-linked inheritance. Okay, so we can see the mapping of the chromosome X here. So chromosome X is the sex chromosome. Okay, the genes are called sex-linked genes because it's located at the sex chromosome. So the characteristic of color blindness and hemophilia are caused by the recessive genes linked to the X chromosome. Y chromosome is shorter than X chromosome and does not contain as many as X chromosome. Therefore, any trait in male caused by either dominant allele or recessive allele on the chromosome X is observed. So sex-linked inheritance ni gen terangkai seks ni maksudnya satu situasi ataupun satu keadaan di mana ada allele yang resesif pada yang menyebabkan color blindness dan hemophilia ni disebabkan oleh resesi allele pada X kromosom. Okey, so dipanggil sex-linked inheritance. Kromosom X ni adalah seks kromosom. Walaupun seks kromosom, tetapi ada juga ciri-ciri di dalam seks kromosom ni yang bukan kontrol tentang sex determination. Dia ada, contohnya macam penglihatan warna kita terletak pada kromosom X. Okey, blood clotting factor kita terletak pada kromosom X. Okey, tapi bila resesif allele pada X kromosom, ia menyebabkan keadaan di mana abnormality kat sini lah, color blind ataupun hemophilia. Okay, so this is the number of genes located in the X chromosome. So kalau Y chromosome is shorter, only 70 to 200 genes. Okay. So color blindness is a condition in a person who cannot differentiate some specific color such as red and green. So color blindness is caused by the recessive allele found in the X chromosome. Okay, and the most people which color blinds are male. In sex-linked inheritance research, X and Y chromosome must be shown by writing the genotype. So the dominant allele is represented by capital letter, whereas the recessive allele is represented by small letter on the X chromosome. So this is how to write down the genotype. For normal person, we have to write the the capital B, okay, so to show the normal male, and female is XB and the capital B, X capital B. Okay, for the carrier, only the female has the is carrier because female have X both X chromosome. So if one of the allele are recessive, so female are carrier. Okay, and color blind for male, bila ada recessive allele B kecil, so dia adalah color blind. Untuk perempuan, kedua-dua adalah allele recessive, baru dia akan jadi color blind. Jadi untuk sex-linked inheritance, hanya perempuan sahaja akan jadi carrier. Okey, hanya perempuan saja akan carrier sebab perempuan ada dua seks kromosom X. Tapi kalau lelaki ada satu saja allele yang resesif, terus dia akan jadi color blind. Kalau female kena ada dua allele resesif, baru dia akan jadi color blind. Jadi most people with color blind ni adalah male. Okay, so this is the example of Ishihara test plate to determine the color blindness. Okay, either we are normal or color blind. So this is the normal vision color, Newton color blindness, proton color blindness, and achromatopsia color blindness. Totally color blind. So this is the schematic diagram for color blind. Okay, father normal eyesight, normal. Mother normal eyesight, carrier. So genotype for normal is X X capital B Y for father, normal, and mother is carrier X capital B X small b, carrier. So after meiosis, father will produce XB, okay, B besar, Y, and mother will produce XB besar, capital B, and X small b. Okay, random fertilization, so it will produce child genotype X capital B, X capital B will produce girl normal, XB B besar, and XB kecil we produce girl normal, carrier. Okay, and this one is X capital BY is boy normal, and this one is we produce genotype X small b and Y, boy with color blindness. So the phenotypic ratio, okay, getting the normal girl is 25%. Okay, normal daughter 25%, normal son 25%, and normal with color blind is 25%. So the hemophilia is the another example of the sex-linked inheritance. So hemophilia is a condition which blood cannot clot in normal circumstances due to the lack of blood clotting factor. This can result in excessive internal or external bleeding, which may be fatal. So hemophilia is due to the presence of recessive allele on X chromosome, same as colorblind, which cause the male to be hemophilic. The female which only the hemophilic if both recessive allele are present on the both X chromosome. Okay, so same as colorblind. So we just change the letter. We hemophilia H. We can use H. Okay, normal male, X H besar Y, female here, and then this is carrier. Okay, only the female carrier the hemophilia, and the hemophilic person. When the recessive allele on the X chromosome here. So this is the condition of normal blood vessel. Okay, and then when for the healthy, we clot the blood the mechanism of blood clotting factor, but for hemophilia unable to clot. So this is the schematic diagram for hemophilia. Okay, normal father X capital HY. Okay, normal carrier, normal female carrier. Okay, but carrier X. Okay, X capital H, X small h. Okay, meiosis. This is the gamete, and the fertilization we produce here. Okay, girl normal carrier, boy normal, and the boy hemophilic. Okay, so the ratio is 1:1:1:1. 25% girl normal, 25% girl carrier, 25% boy normal, and 25% the boy hemophilic. So ability to roll the tongue and the type of earlobe is another type of the inheritance trait in human. Okay, ability to roll the tongue, the type of earlobe. So is two characteristics can be inherited from parents according to the Mendel's law. So ability to roll the tongue is dominant trait, and free earlobe is dominant trait, whereas the attached earlobe is the recessive trait. So for inheritance, we can use family pedigree to show the can analyze the and investigate the inheritance of human characteristic. Here the symbol used in the pedigree. Okay, this is symbol for normal male, normal female, hemophilic male, hemophilic female, or female carrier. So family pedigree is flowchart through a few generation to show the ancestral relationship and inheritance of characteristic from ancestor to individual in the present generation. Okay, family pedigree ni nak menunjukkan pewarisan daripada keturunan mana yang yang seseorang tu boleh dapat hemophilia. Contohnya hemophilia daripada keluarga ni, okay, datang daripada carrier, daripada generasi yang kedua. Analysis of family pedigree enable to genetics to predict an inherited characteristic of interest and also to identify the feature of dominant or recessive genes. Normally, dominant genes appear in every generation, whereas a recessive gene is probably hidden in certain generation. Okay, so selalunya macam tu lah. Normal dominant selalunya ada dalam setiap generasi sebab dia dominan. Kadang-kadang recessive tu akan ada, mungkin certain generation dia akan hidden. Okay, that's all. Thank you.